Hi everyone, I'm Yanan, a PhD student from Civil and Environmental Engineering. Today my topic is about the investigation of PFAS transport in soil zone. I think as is the last section, this section is about PFAS. I do not to talk about too much basic knowledge about PFAS, but PFAS is everywhere and it has hydrophobicity and hydrophility properties. And from this picture, we can see the source of PFAS are almost everywhere. PFAS emission may occur during the use of PFAS, the degradation of precursor, and the disposal of PFAS-containing products. The PFAS released in the environment will transport among the air, water, soil, and sediments, and PFAS in the environment can enter human's body by food chain and food net, which will affect our health, our body. Well, PFAS has been widely used since 1950s, and after more than 70 years, we found thousands of PFAS. According to the knowledge we have now, PFAS can be divided into the polymer PFAS and non-polymer PFAS. Sure, the transportability of non-polymer PFAS is better than the polymer PFAS. This study is focused on the non-polymer PFAS. Well, soil and sediment represent an important pathway that human exposure to PFAS. After PFAS released to the soil sediment, PFAS behavior such as transportation, precursor transformation, adsorption, and evaporation, that they make it complex to describe the PFAS underground transportation. Model learning is a good way to estimate PFAS behavior, while it needs a lot of data to train and test the model first. There are some previous studies try to set up a model and use to describe PFAS behavior, while as we talked just now, high resolution data will be needed to test and improve the model. So a high precision vertical profile field PFAS concentration is urgently needed for further research about PFAS transport vertically. Well, this study is focused on PFAS transport and retention in the unsaturated zone. We collect a sample from a PFAS contaminated vadose zone, and the sediment were collected every half feet from two sides to get a high resolution vertical profile field PFAS concentration. Let's see the picture on the left side. The vadose zone or unsaturated zone we talk about here mean the area that below the ground surface and above the water table. At those sites, the water table, at those sites that we ice sampling, the water table there is about 12 feet. To avoid the groundwater fluctuation effect, we collect samples from 0 to 9 feet, and all the sediments we used were saved and dried before we use. Well, we set up an extraction method to extract PFAS from sediment. For the PFAS quantitative analysis, we added EIS is extraction internal standards to the sample, and then equilibrate for more than 30 minutes. Then we use the methanolic ammonium hydroxide to extract the sample. To extract completely, we extract every sample 3 times and then combine the liquid together. After the extraction, the PFAS concentration in the liquid is low, and there is impurities in the combined liquid. To purify and concentrate the sample, SPE, the solid phase extraction, was used. Then the liquid sample goes through SPE cartridge, PFAS will absorb on the cartridge, and impurities will flow out. After that, you use some you use some elution to wash the PFAS out. Then the PFAS solution can be measured with LC mass. So you can get the PFAS concentration in the liquid, and after calculation, you will know the PFAS concentration in the sediment. And in this study, the method detection limit is around 0.04 nanograms per gram. Well, to detect or to test the recovery of the method, I used 10 common PFAS to test. I spike the mix of the PFAS solution to ultrasound and then extract it out to see if the method works. From the result, we can see the recovery of most PFAS is more than 90%. There is a minor loss of PFBA and PFHXA was observed, but even with the loss, their recovery are still more than 75%. That means the method works and we can use it. So before we extract the sediments, we characterize the sediment sample with XRD, XRF, and FTIR. We can see the peak of, from the XRD result, we can see the peak of dolomite and quartz. I'm not sure if you can see it clearly as the screen is not so big, but I can see the red marks belongs to dolomite and the green marks belongs to quartz. And from the result, we can see the sediments is heterogeneous. The dolomite content of the sediment from the surface layer is higher than that from the deep layer. And from the FTIR result, we can see the peaks belongs to carbonate and the silica oxygen bond. The XRD and FTIR results proved that the quartz and the dolomite are the major minor minerals in the sediments. From XRF result, we can also say the result makes sense with XRD and FTIR result. Dolomite and quartz are the two main minerals in the sediments we used here. Well, in this study, when we try to extract PFAS, we targeted 12 PFAS, except Genex. The other 11 PFAS results are shown here. We didn't find a PFAS concentration in the sediment. I think maybe because there's no, or maybe because the concentration is too low to detect. For any of the other 11 PFAS shown here, their concentration in the sediments, we can see the concentration increased first and then decrease when we increase in the sampling depth. From the concentration, you can see the x-axis. We can see the peak concentration of like PFOS, PFOA, and PFHXS are higher than other PFAS. Another thing we can recognize is the position of the peak, the highest concentration position of each PFAS. Here we can see the peak position of long-chain PFAS, such as PFOS, PFNA, and PFDA. Their peak show at a shallow layer compared with the short-chain PFAS like PFBA, PFBS, PFPA. The short-chain PFAS, their peak are shown at a deep layer. In deep layer, like 7 to 9 feet, the long-chain PFAS concentration was super low there. And that show in both sides, also in this size. We calculate the property of each PFAS in each sediment depth. The result shows in the figure. We can see when the depth is shallow, the main PFAS was the long-chain PFAS. And when the depth is increased, the short-chain PFAS become the main components there. When we analyze the distribution of each PFAS in each layer of sediments, we found that for short-chain PFAS, most of them accumulate in deep layer, like 7 to 9 feet, compared to the long chain PFAS, which accumulate at 0 to 2 feet. So that result show in both sides. We can see different PFAS has different transport behavior. When PFAS has same functional group, the longer chain PFAS has a shallower transport depth. When PFAS has the same chain length, the carboxylic acid has a deeper transport depth than the sulphonic acid. We know it's in the same site, same environment condition, but different PFAS show different transport behavior. So I can suspect that due to the PFAS has different property. As we mentioned at the beginning, the hydrophobility and hydrophobicity are important property of PFAS. In this study, we use the octanol-water distribution coefficient. We call it, when you work in lab, you call it log DOW. We use that to explore the relationship between the PFAS property and PFAS vertical transport depth. To explore the relationship, we selected half of the total PFAS and calculated the corresponding transport depth. Plot the depth and log DOW in a bar plot. We can see in both sides, the log DOW and the PFAS transport depth show a negative linear relationship with each other, which means when the value of log DOW increased, the PFAS transport depth decreased. We know the value of log DOW increased means the hydrophobicity of PFAS increased. That indicated that the PFAS hydrophobicity plays an important role in PFAS vertical transport process. So I think that's what we analyzed from the data we got. Sure, as we said at the beginning, the model learning is a good way to describe the PFAS transport at the ground. So Hydros 1D was used to simulate the PFAS vertical transport profile. The main parameters we adjusted in Hydros 1D are listed on the table. And the result we got up to now is also shown here. And it seems the sampling size, it was polluted by the PFAS for more than 40 years, seems.
Minutes remaining.
The high precision data was used to train and test will be used to train and test the model in the future, and the model will be used to simulate the PFAS transport tendency in the future. This work is still ongoing. I think this summer we will get more information. I think that's all what I want to share today. At the end, I want to say up to now we got a high precision vertical profile, and we know the PFAS retention within the widowed zone depend on the widowed zone depth and the PFAS structure. And for given PFAS, the concentration increased first and decreased when we increase the depth. And the PFAS migration rate was negatively related to the octanol-water distribution coefficient. And at the end, I want to thank my advisor, my lab mate, and thanks for listening. Thank you. I'd like to take any question if I can. Hopefully I can. Thank you.
We have about 30 seconds left, so I'm going to recommend that we ask questions on Hoover.
Sorry about it, but if you have any question, I think I can answer from over.
Yeah, sounds good.